in the spring college, so it's a bit of a different experience. And as was said, the plan for this week and next week, actually it's going to be 2 people. So I'm Ariel Amir, first name, last name, and don't be confused, Amir Erez, first name, last name. So I will talk this week about microbial evolution. So today I'll talk about Luria-Delbruck experiment, which is also about evolution, but in a sense it's the start of our understanding of evolution. We know, well, okay, I'm not going back to Darwin. Let's say it's the start of quantitative evolution, and you will see what I mean today. And then later in the week I'll talk about size control, which I think is related in a sense to some things you heard last week from Jacopo Grilli. And if maybe we'll get to gene expression and protein production, that will be even more connected to the first course you had on quantitative physiology. Okay, so that's the plan. And of course, feel free to stop me at any point. If anything is unclear, if I'm going too fast or too slow, just let me know. Okay, okay, so today we're going to talk about microbial evolution. And you've heard a little bit, so you already know what these are, right? What are these? What am I showing you? Bacteria. In this case, it's B. subtilis. It's a soil bacterium. But actually, the main hero of this story is a different bacterium called E. coli that we all have in our guts. And it's sort of the hydrogen atom of quantitative physiology. And it has been like that for decades. So there are many, actually I asked ChatGPT a couple of weeks ago how many Nobel Prizes we're giving to hydrogen. I don't know, there are 5 or 6. How many to E. coli? It's a similar number. So in both cases, there has been a lot of understanding coming from the study of this particular bacterium. What's special about it? The hope is not much, right? The idea of working with a model organism is reflected in this quote by Monod, who got a Nobel Prize for studying E. coli. Anything found to be true of E. coli must also be true of elephants. Okay, so the hope is that the principles that you learn from studying E. coli will generalize. And a lot of times they do, and the example I'll show you today, it was the experiments done on E. coli, and today we know that they're far more general, and it'll be clear to all of you what I mean today, that the principles that drive evolution are the same whether I'm talking about R evolution, E. coli evolution, or COVID evolution, right? Mutations, random mutations, the genetic code is the same, so the principles are the same. Now, of course, some things E. coli is not going to be good model organisms for example, developmental biology, right? We have tissues, different cell types. E. coli is probably not such a good model for that. But many other things, it is. Okay, so don't take this quote too literally. But it's still an important model organisms. And for those who like this sort of thing, I highly recommend. This is a popular science book. Not a single equation in it, but it has a lot of beautiful science in it. It's called Microcosm by a fantastic writer called Zimmer. And all the book is about discoveries that came from studying E. coli, including things from the last 20 years. The field is moving quite fast. The book is from around 2010, so it's a bit outdated, but still there are decades of research in it. Very, very enjoyable read, including what I'll tell you today. They also will talk about, without equation, we'll do the kind of gory details in the equations, but they will talk about the Luria-Delbruck experiments and other related things to that. Okay, so we can learn fundamental biology from studying E. coli. There is this quote, this book. Today we're going to talk about the role of mutations in evolution. And the one last thing I want to show on this slide is that I chose this example, and I call the course Physics of Bacteria. Okay, and I should say next week, Amir is going to continue still with microbes, but talk about ecology. Okay? For me, it's all going to be about single cells that are not interacting. But of course, there are many levels to this problem, and bacterial cells also interact both with their own species and with other bacteria species and with their host and so on. So Amir is going to talk about ecology. Okay? But in the example that I took, single cell, I still call it physics of bacteria, but it's different physics than the sort you're used to. Well, maybe by now you're used to because you heard Jacopo speak about quantitative physiology, but there are no forces, there are no Maxwell's equation, okay? So why do I call it physics? Because it's the approach, okay? The approach of taking a complex problem, and I think this course is called Physics of Complex Systems. I think this is a complex system. It's hard to argue that take a single bacterium in your gut. It's hard to argue that it's not a complex system. It's a hugely complex system, and we're scratching the surface of understanding it. Why is it so complex? Because it has been evolving for billions of years, okay? There is a lot you can achieve in a couple of billions of years. So it's a hugely complicated system, okay? And to understand it, or to try to understand it, we make simplified models that are usually coarse-grained. We don't try to capture all the details of every protein within a bacterium and how it interacts with every other protein, because it would be a useless, futile mission, right? There are thousands of genes, and to account for how all the genes interact with every other gene, it's going to be impossible, okay? So what we try to do is the opposite, try to make minimal models that still capture an important feature of the problem. And that's what we'll have today. We'll talk about evolution, and I'll show you a hugely simplified model that still makes quantitative predictions that we can test, falsify, and so on and so forth. Okay? So that's why this is an important example that physics doesn't have to have physical forces or Newton's or Maxwell's equation. It can be the approach. And I think it's not a coincidence that in what I'll start with today, it's called the Luria-Delbruck experiment. Luria was a biologist, and Delbruck was a physicist. Okay? He actually started working on quantum mechanics. He made some important discoveries there. There is an effect named after him in quantum mechanics, Delbruck scattering, that people still work on. But then he switched to quantitative biology, made a lot of important contributions across fields on the border of biology and physics, because he brought with him the physicist approach and the quantitative approach that was very important. Okay, so before we describe the experiment, this might be redundant with what Jacopo already told you, but I'll repeat it nonetheless, and I'll just take a couple of minutes. And again, feel free to tell me if I can skip this or if you have any questions. So here is an E. coli, different time, so it's growing. Let's say it's a micron wide and a few microns long, and it doubles itself and then divides into two daughter cells. So asexual reproduction. One cell becomes two cells in a time that can be as short as 20 minutes if you give it good nutrients. And I saw in your exam here that you spoke about nutrient quality and things like that. So you should know what I'm talking about. And here is how it looks like. So on the left, here is E. coli growing on agarose pad. So it's basically a surface where the food is inside this gel on the surface. And you see the cells doubling and dividing, creating this nice colony. So this is what we're talking about. But we're not interested in interactions between cells. We're talking about single cell behavior during this week. Okay, next week is going to be different. It is going to be about interactions. Okay, here is another, here is another example of a microbe, but not a bacteria. So this is yeast. Okay, yeast is important for making bread and making beer. And you can see here the cells are doubling with a doubling time, mass doubling time of about 90 minutes. And this is a fluorescent protein that is marking the division. And sort of similar thing is happening. And a lot of the principles also will apply to things like yeast growth. And then the last one I'm going to show you here, it's called Helobacterium salinarum. So you might think it's a bacteria, but then a couple of decades ago, people understood that it's not, and it's an archaea. Okay, archaea are the third domain of life. There are three domains of life: bacteria, eukaryotes, and archaea. And they're often extremophiles. So this is a microbe that can grow at very high salinity concentrations, places like the Dead Sea in Israel. it grows much slower. So here the mass doubling time is 4 hours. But other than that, it looks a lot like the E. coli video on the left. So you see it doubling and dividing, doubling in length and dividing. Okay? And we'll talk about some commonalities of these microbes in the second topic of cell size control and how cells know when to divide. Okay? So how do these microbes know it's time to divide and they should split into two daughter cells? This we'll talk about in the second part. Any questions so far? Yes? Okay, good question. So E. coli is very, very good at dividing symmetrically. So if you look at an E. coli cell and you look at the size, let's say it decided to divide here, you look at the size of the daughter cell divided by the size of the mother cell. Okay, so you get a fraction that's a Gaussian centered around half, okay, with a standard deviation of a few percent, let's say 3 to 5%. So it's remarkably accurate, and there is a whole, I could have spoken for the whole week about the physics behind that. Okay, so there are at least two mechanisms that help it divide symmetrically. Can anybody guess one of them? Yeah. Okay, so you know too much, but like that's not called guess. It's like knowing the answer. But like there is one mechanism you can guess, which is what do these bacteria have to have in them, and it's always going to be more or less the same, and it's related to today's topic as well. So what do they have? We all have. Yeah, so they have a genome, right, and DNA, and the cells might look a bit different when they divide, bigger, smaller, fatter, but you need to have the same DNA because that's the genetic information encoding everything you need, okay? So in bacteria, such as E. coli, the DNA, the information is encoded on a chromosome that's circular. It has the origin of replication, and it has the terminus, and you duplicate it. And we'll talk about this in the second, maybe in the second topic. So I've got like my chromosome, and it looks like this tangled mess because it's not a nice, it's very long. If I burst the cell length is a few microns. The DNA will take up about 1 millimeter. Okay, so it's huge compared. So all of this is highly compacted inside the bacterium. And there is another one here. And they're identical, right, up to potentially some very rare mutations that we're going to talk about today, irrelevant for this. So it's essentially two identical springs pushing against each other. And it turns out that you cannot divide. The division machinery is set up in such a way that it's not going to divide over the chromosome. That will be detrimental. If you cut your chromosome when you're dividing, it's game over. There's no point. You're dead. Okay? So what are the places that you can divide? Anybody? Where can I divide then? I cannot divide over the chromosome, so what's left? The middle. Okay, so that's one. And what else? The poles. So these are called the poles. Okay, and so I can divide here and here. If I divide here, it's also game over for that cell. Okay, so I really don't want to. I will lose the whole point. I spent all my energy doubling everything, and now I kind of divide in two, and I only have one happy cell. This one is fine. It's got, intact chromosomes, even two of them. That's going to survive. But this one is just a sac with no DNA. It's going to die. Okay? So cells grow to great length not to divide at the poles. How do they do it? By a set of proteins that I'm not going to talk about, but were mentioned here, called the min proteins. Okay? So there is min C, min D, and min E. And these are a set of proteins that work together in a sort of crazy way that creates, through the feedback between the proteins, waves. So if you measure these proteins, you know that you can label proteins. Did you talk about fluorescent labeling? You steal a gene from jellyfish, you stick it inside the E. coli, and then every time you have, say, min C, you'll see a bright spot. You can measure it under microscopy. Okay, so I can measure the proteins, and what you find in E. coli is that these min proteins, if you look at their concentration, it's actually a traveling wave that goes back and forth across the cell with a time scale of about 30 seconds. Okay? So what do they gain? And now I have this protein creating these waves back and forth in the cell. If I have this thing, and it turns out the min proteins are also suppressing the division. Okay? So the min is set up in a way that doesn't allow the cells to divide. If I have something moving back and forth, where is it going to have the maximal blocking effect on division? Where it spends the most time. Where does it spend the most time? At the poles, because it momentarily stops there. Okay? So that's where it's going to block division. Where is it not going to have such a big effect? In the middle, because it goes through quickly. So all of these chemical oscillations, all this crazy system is built to suppress divisions at the poles, okay, and allow the cells to get this very high accuracy of dividing in the middle. We can make mutations in bacteria, so today we can control them. So if I make a delta min, I forgot which one, let's say C, so I knock out this protein, I get rid of it, the cell is going to still be pretty good, but now what am I going to get? Polar divisions. So suddenly I have a population that wasn't there before that divides and creates what we call mini cells, which are basically cells without any genetic information, which can be useful for us, for investigating this mechanism. Okay, so again, it's a prediction, if you want, of this picture. If I get rid of the min, I will get divisions at the pole, and that's indeed what you see. Okay, so that was not something I plan to talk about, but please ask questions, because I think it's more fun this way, and we can learn more. Any other questions? Okay, so if we look at this, this is the phylogenetic tree of life. So what do we do? Basically, by looking at the sequences, we can infer what happened during the course of evolution over billions of years. And the picture we have today is that bacteria branched from eukaryotes, which includes animals and us. So again, to get a sense of humility, this is us, this is the animal kingdom, and we are between slime molds and fungi. Okay, this is where we are. Bacteria are here, and archaea, which were only understood several decades ago, so they're a fairly new discovery. They branched off from eukaryotes, and in a sense, they share some commonality with eukaryotes and some with bacteria. So their morphology, they look a lot like bacteria. People thought that they are bacteria, but actually when you look into things like their DNA replication machinery, it's more similar to eukaryotes than to bacteria. Okay? And I showed you on the previous slide examples of microbes from these three domains of life. So these are called the domains of life. Okay, so these were the budding yeast, H. salinarum, and E. coli. Okay, so now finally we're going to talk about the Luria-Delbruck experiment. And I'm going to describe the experiment. It's very simple. And you can do it in any university today in a day. It's not like you need sophisticated machinery or anything like that. And the experiment goes like that. You start with a handful of bacteria, let's say 100 bacteria in the original experiments, and you put them in some nutrients, so you grow them up to about 10 to the 8 cells. So from 100 to 10 to the 8 cells. Now I expose the cells to a virus. Just like we have viruses, there are viruses called bacteriophages that attack bacteria. And this virus, it looks like the vehicle that lands on Mars, and it basically lands on the bacteria and kills almost all of them. Okay? And now I want to count how many of the 10 to the 8 bacteria survived. Okay, so this was done in around 1940. So the year is 1940. I have all my bacteria. I grew them up. How do I know how many survived? What do I do? Any suggestions? Well, 1940. I mean, low tech. Count what? Okay, so I take my culture. I mean, I'm slightly simplifying, but basically I take my culture and I can spread it on one of these agarose pads and wait a couple of days. And what will I see after a couple of days? Colonies growing, right? So it will look something like this, like on this Petri dish. And I know that every colony came from a single cell. So I can count how many cells survive. So when you know, general lesson, even when you read a modern paper, don't just take the results for granted. Ask, you know, you don't need to know which microscopes they used necessarily, but you want to understand how they measured what they measured. What's the principle? Okay? Yes? It could be. It could be. But, you know, let's say I'm a Luria, Salvador Luria in the year 1940, and I want to know, is this a negligible effect or not, merging of colonies? What would you do? So one thing you can do is take the culture, and before you plate it, you can dilute it, say, by tenfold. So now if this was a big effect, you should have a dramatic effect on the number of colonies because now you'll get less merging by a big factor. So we do this, you know, Gedanken, nothing happens. So then I know, okay, so this is a negligible effect, I can forget about it. So it's a good point, it's a good comment, but we can test and make sure that this is not an artifact we have. And actually, Luria spent a long time making sure that their protocols are robust to these technical details, let's call them. But definitely we should be mindful of that. Other questions, comments? Okay, so here is the output of the experiment. On the x-axis is the number of survivors, and on the y-axis, it's the number of experiments during which this happened, so the fraction of experiments. So this is a histogram. Everybody okay with histograms? Okay. So in 35% of the cases, more or less, it was a very boring experiment. Nothing survived, 0. So I plate, I see no colonies. Okay? And then in, I don't know, 3%, I get, look at the black. The gray dashed one is going to be the theory that we're going to get to. But this is the experiments. So 35%, nothing happens. Few percent, I get 8 survivors. And then there are about 30% of cases where I get more than 9 bacterial colonies emerging, 9 survivors. And in fact, there are cases where you get 200 survivors. Okay? So if the year was 1943 and you just did this experiment, how would you feel? Surprised. Okay, why surprised, or?
Because we were expecting that the number of, like you have this histogram to just go down, and then suddenly.
So that's not why surprised. So here it's not 9, it's everything other than 9. So there is some kind of tail here. Okay. And I just dumped all of the tail, I lumped it into one bin, and I put it there. So that's not the surprise. But let's say this is what you get, okay? So something that decays, you know, like this. Still, what do you think? What do you learn from this? Yes? Yeah, so like I just did an experiment, and it's not reproducible. On Monday, I got 0, and on Tuesday, I got 9. And every time I do the experiment, I get something else. And you can say, that's not what an experiment is supposed to look like. Okay, so Luria spent a long time testing, you know, maybe I'm not controlling temperature, maybe I'm not controlling humidity, maybe, you know, where is the artifact? Where is this coming from? Okay, and it took time for them to understand, yes? One is theory, one is experiments. We didn't, so just ignore this one, look at the black. Okay? So it took them time to understand that this is the most important result of the experiment. The fact that it's so variable and not reproducible is the main result. And I think that most scientists, if they get a result like this, would throw away the experiment, declare it a failed experiment, and go home. And they decided to not do that and understand it, and they did, and they got a Nobel Prize essentially for understanding this plot. Okay, so that's also a good lesson about persistence. Yes?
So the reason that each experiment like different results in a different day, are we like talking about stochasticity here?
We are talking about stochasticity, and we're going to quantify the stochasticity now. Okay? So wait with this, and you'll see why this information is useful and how we can quantify the variability in these plots. Because we don't expect anyway in biology things to be, we don't expect 55 survivors every day, right? That would be also crazy if I always get 55. It doesn't make any sense. So we expect some variability. So it's not just about variability, yes or no, it's about quantifying variability. And to quantify variability, we need the right metric. And so soon we're going to build a null model that is going to be wrong, but the null model will show us why the right metric to analyze this data. But before I say the answer, can anybody give me any metric? It's not, I don't expect it to be the right one, but any metric to measure variability from this data. Let's say I want to quantify variability of this plot. What can I do? Anything. No, I'm asking about data. It's a question about data. Here is data. How do I quantify how variable it is? Give me any metric to quantify its variability. Okay, so you can say I can fit this to a power law and get the power. That's good. It's a bit, maybe I don't have enough data to fit a power law here. I want something even simpler. But it's good, and by the way, we'll see it is supposed to have, if I had a huge number of experiments, it is supposed to be a power law. That's supposed to be a power law toy with a very specific exponent, so it is a good idea. Yes? Perfect. Perfect. Okay, so that's even the right one, so we'll see why the ratio of the variance to the mean is the right way to quantify variability. But of course, before we do the calculation, you could have said maybe it's not mean and variance, maybe it's the standard deviation compared to the mean. Standard deviation divided by the mean is called the coefficient of variation. It's also very commonly used to quantify variability. Here it's not the best metric, as we'll see, to quantify variability. But a priori, it could have been a coefficient of variation as well. Okay? So sometimes you need to go to, in some theory of glasses, you need to go to a fourth order higher moments, but usually the lower is better because it's more reliable. You can measure it from the data. It's hard to measure higher moments from actual real data. Okay, but variance is okay and mean is okay. Yes? Just for presentation, because otherwise you'll see, like, if I continue this to 243, it'll be somewhere where the construction is happening. So when you calculate the variance to mean, you take all the data. You don't need it's just a presentational simplification. It's not. I'm not throwing away any data or anything like that. Okay. So yes.
Sorry, when you say that the experiment is not replicable, you mean that one result is not replicable, but the average of all the results will give the same distribution?
Not really, because the experiment, I do it on Monday and I get 0 survivors. I do it on Tuesday, I get 15 survivors. So are you saying if I average enough experiments? Yeah, maybe, but I have let's say I did 300 experiments. What do? I take the first 150 and the second 150? No, okay, good. So you're saying the histogram is reproducible. More or less. You can see it's kind of quite noisy. So probably if I do the whole 300 experiments again, I will get a similar histogram, but not identical. But yeah, good point. The statistics are reproducible. Okay, so let's dive into it. And here are the two theories we're going to discuss. And now remember, the year is around 1940. Nothing is understood about evolution. Okay, the double helix, not discovered yet. We don't know much about the genetic codes, certainly not about mutations. And there are two hypotheses on the table. Okay, who here has heard the words, well, Darwin, I'm sure you heard about. What about Lamarck? Some. Okay. So Lamarck, maybe you know the kind of story from high school, has a theory that, if you look at giraffes in the savannah, they stretch their necks. And because they stretch their necks, their offspring is going to have longer necks. Okay, so you kind of try to do something, and then your offsprings will be adapted because the parents were adapted or tried, then the offsprings will become adapted. Okay, so that's a Lamarckian picture. Okay, so what's the analog here, which sounds pretty reasonable? Okay, the analog is I'm a bacterium. Okay, the singular form of bacteria is bacterium. So I'm a bacterium, and I'm exposed to the virus. Okay, so I've got these things coming and trying to kill me. And there is some probability P that I survive, and a probability 1 minus P that I die. Okay, and this P is going to be very small. We know it's very small because out of 10 to the 8 cells, most of the time, all of them die. So P has to be of the order of 10 to the minus 8 or something like that. Sounds perfectly reasonable. And by the way, once I survive, what happens to my offsprings? The cells, all the cells I produce, this one, and then divides into more and more. They all survive. And that's testable, by the way. I take the again, how would I test this? Very easy. I take one of the colonies in the experiments. Okay, I grow it, and I see that everything survives. That's true. Okay, so in the experiments, once you survive the virus, all your children, all your offspring survive the attack. So this sounds like a perfectly reasonable model. Okay? Note that it's the Lamarckian picture, because in this case, I'm attacked by the virus, I try to survive, and either I adapt or I don't adapt. If I adapt, my children are adapted. Okay? So that's one model, which sounds, again, it doesn't sound very far-fetched. It sounds perfectly reasonable. Probably if I had to force you to write a model for this, this is the model you'll come up with. Okay? The important thing is that in this model, each one of the 10 to the 8 cells is essentially has an independent chance of surviving, right? Each one, I roll the dice separately. For each bacteria, there is a probability P. It's a coin flip. It's a biased, it's not a fair coin. It's a coin that has a very small chance of surviving, but I flip the coin independently for each one of the 10 to the 8 cells. This is a key point. Okay? What's the second picture? The second picture is the picture that today, especially after COVID, you all know to be true, right? That if this is the, say, one bacterium becoming 10 to the 8, doesn't matter that in the original they started with 100, it's a detail, okay? I grow this tree, and there is a chance to get random mutations well before I'm exposed to the virus. So the being exposed to the virus is irrelevant. I get the mutations randomly all the time, right? Every time a bacterium duplicates, there is a chance that it gets point mutations, and some of these point mutations might make it resistant to the bacteria, to the virus. By the way, same thing with antibiotic resistance. You can spontaneously get antibiotic resistance just because of these random mutations. Okay? What's the difference? Sounds like a very similar picture. Well, the difference is this. If you look at this drawing, you see that on the left, you get a survivor here, you get a survivor here. They're independent of each other. Here, if the bacterial as a population got lucky, you might get this mutation early on in this tree. What happens if I get this mutation early on? All of this subpopulation here becomes resistant to the virus. Question?
I have a weird question. Can you undo a mutation with another mutation?
Yes, but it's negligible here. So if you calculate the kind of probability of a back mutation, it's going to be completely negligible. So you can forget about this scenario. In other cases where if the mutation rate was very, very high, then yes, you should worry about back mutations like that. So it's a good question. Other questions? Okay, so I want you to kind of, first of all, before we dive into the calculations, we had a professor at Weizmann when I was doing my PhDs that said a calculation has to be preceded by an idea. Okay? We don't just calculate. We first need to think and have a picture of why we are calculating what we want to calculate. So we understand variability is key. And we have this intuition that here it's going to be maybe more variable than here. Maybe you have that intuition. If not, we'll do the calculation. You'll see. But the reason is that here there is a chance of getting suddenly a whole lot of resistant bacteria. Why? When the mutations happens early on. And then suddenly you hit the jackpot and you get a whole lot of resistant bacteria. So the story is, I think it's true because I think that Luria wrote it in his biography, that he went to a party and they were playing this coin games where usually you get nothing, right? And then once in a while you get a huge amount of money. Okay? So then it gave him this idea, and Delbruck did the proper calculations, and they saw that it's true. And that's the sort of calculations we'll do now. So sometimes this distribution is called the jackpot distribution, the one corresponding to the scenario on the right. Okay? So we're going to get very different distributions here and here. And they're going to be characterized by a different ratio of the variance to mean. Okay? So all of this was an introduction. Now we're kind of ready to do the calculations. And let's start with the easy case, which is the case on the left. Okay. So again, not to confuse you, there are two scenarios. Now we're talking about the Lamarckian case, where every cell is characterized by a parameter p, the probability to obtain resistance that will then be heritable. Okay? And let's say I have at the end this n equals 10 to the 8th cell. And let's calculate two things. First of all, what is the mean number of survivors? Pn. Okay, super easy. Okay? They're all independent, nothing to it. Okay? Now, let's look at the variance. Now, the number of survivors, I can think about every cell as having a random variable associated with it, which is, say, 1 if it survives and 0 if it dies. Okay? Then the total number of survivors, okay, is simply the, let's call it S, is the sum of Si. I goes from 1 to n, where Si is this random variable. Sorry, maybe I put notes online, so maybe I'll stick with the same notation I use in the notes. Okay, so I called it X. And it's the sum of these Xi's. Why is this good? These are independent variables. So I know that the variance of X is simply n times the variance of one of these variables, Xi. Okay? So what is the variance of Xi? Okay? That's super easy. It's P times it survived. So I need to take 1 minus the mean, which is P, right? And square it. Plus 1 minus P, I didn't survive. If I didn't survive, my variable is 0, so 0 minus P squared. Is everybody okay? I'm just calculating the variance of a Bernoulli coin flip. I have a random variable that is either 0 or 1, 1 with probability P, and the mean is P, and so this is how I calculate the variance. Everybody on board? Okay. So now simple algebra will give you P times 1 minus P. Okay, you might remember even this result, that this is the variance of a Bernoulli coin flip. P is tiny. So this is approximately, to a good approximation, simply P. Okay? So this is a very important thing we have here. We just understood that for this Lamarckian model, the mean is p times n. And what will be the variance? Approximately, to an excellent approximation. What's that? Wait, we just calculated it. So why? So something is unclear. This is a random variable. It's a sum of independent variables. Each independent random variable is a coin flip. Okay? We just calculated the variance for a single coin flip, and we got it's about p. So what's the variance? P times n. Okay? So we expect the variance to be equal to the mean. Now, for the data I showed you, if you calculate the variance divided by the mean, you get something of the order of hundreds, say 300. So you can already rule out this model. Okay? But I want to actually say a few more words about it. Just because we can and it's fairly easy, we can also understand that the shape of the distribution in this case is supposed to be very different. Yes? No. So actually, great point. So this calculation tells us variance to mean is the best metric. Why? Because we have two models, and in one of them, the variance to mean, not the square root of variance, but the variance to mean, that's what's predicted to be 1. That's a prediction. It's a wrong prediction. It turns out to be wrong. But if I do square root, it will be different, right? It's cleaner to work with the variance to mean. Then this model predicts 1. The other model, we will see, predicts a huge value. So then I can rule out this model, and I'm left with the other model. Okay? But let's kind of be greedier. Let's say I don't just want to look at this single number. Let's say I want to ask, within this model, what would be the shape of the distribution? Okay? You have all the tools for that, right? So can anybody think for a second and tell me what's the form of the distribution we expect in the case, which is wrong, we know today is wrong, that the Lamarckian picture is correct? So what kind of distribution do we get? Or let me ask you it in this way. What's the probability to get precisely x survivors? Okay, x can be 0, 1, till 10 to the 8. What's the probability to have precisely x survivors in this Lamarckian picture? Choose. You mean this, right? Yes. So n choose x is the number of ways I can choose x out of n. It's n factorial divided by x factorial, n minus x factorial, right? Now, if I chose these x, these need to survive, and the rest need to die. What's the probability that these survive? P to the power of x, and the rest need to die, 1 minus p to the power of n minus x. So this is called a binomial distribution. So we know that this is the result. But here, we can actually make simplifications. Why? Because we know that x is much, much smaller than n. We never care about the case of getting 10 to the 8 survivors because it has negligible probability, right? We know that because p is so small. So then we can approximate p of x as, n over x factorial is n minus 1, n minus x plus 1, divided by x factorial, right? But x is so small compared to n that this is essentially n to the x divided by x factorial. Is everybody OK with that? I just replace all these factors by n because it's a good enough approximation. And then I get times p to the x, 1 minus p to the n. Okay? Now I want to make one more approximation. You'll see where I'm going with this in a second. And again, stop me if anything is unclear. So this one I'm OK with. This one I'm OK with. And I want to approximate this thing. Okay, I have 1 minus a small quantity to the power of a large quantity. What can I approximate that with? Exponential, excellent. So there are two ways to do it. Okay, one is kind of, I can write this as 1 minus np over n to the power of n. I didn't do anything, that's exact. But then you remember that the limit of large n, 1 minus x over n to the power of n, is e to the minus x. So this is approximately e to the minus np. And if you want a slightly more systematic way to do it, you can write this as e to the log of this quantity. The log of this quantity is n log 1 minus n, 1 minus p. And now if you have an exponential of n times log of 1 minus something small, you can, what would you do? Taylor expand, right? So you can Taylor expand this log, say, to second order, and then the first order will give you exactly this, and the second order will also give you the correction, so you can know when this is a good approximation or not. So it's kind of useful to at least know how to do this more systematically. Okay? So this is what we got. So at the end of the day, when we put it together, we find that p of x is approximately equal to lambda to the power of x e to the minus lambda over x factorial, where I introduced here lambda as n times p. What do we call this distribution? Poisson. Okay? So we see that in our case of small p, the binomial, which is the exact result, is approximated by Poisson. Now, in physics, you probably know this. Knowing how to make approximations is critical, right? So it's not that exact is better. It's usually the opposite, that if you can simplify in the appropriate limit, you understand better and things are simpler and more useful. Okay? So for the Poisson distribution, if you take this Poisson distribution and calculate the variance to mean, you kind of know the answer already. What are you going to get? 1. Okay? So you can do it. You can calculate the variance of this thing and the mean of this thing. In a sense, we did it in a smarter way, relying on the independence. We proved that it's 1. Okay? So we don't. It's better to do it our way. But if you want, as an exercise, take the Poisson distribution, and you will see that indeed the variance to mean is precisely 1. It's exactly 1 in this case because we already made the approximation of small p. Okay, so it's just when you take this point of a small p. So why did I go through this? Because in principle, you can also take the data and ask, is it well fit by a Poisson distribution? And the answer is no. Okay? But I guess the simplest way is to look at this variance to mean, measure it in the experiment. We have a prediction, it should be 1. Doesn't turn out to be, it turns out to be wrong. Okay? So what we're going to do next is calculate the variance to mean in the other model, the model that we know is true, which is random mutations drive evolution. So again, maybe a couple of years ago, I would have a hard time. Today, you all know about Omicron. You get some random mutation somewhere, and suddenly it spreads over the population because it has a higher fitness and so on. Okay? So within our model, this is the picture. We can get a mutation. So we still have a single parameter to characterize things, but now it's not the probability to survive at the end. It's a probability to get at any replication event a mutation that would make me resistant to the virus. Now, I want to stress a few things. First of all, I have a long genome. Okay? When I talk about mutation, it can be actually maybe a dozen different mutations at particular places that give me resistance to the virus. So there are 2 points. First of all, mutations happen all the time. So I'm not talking about any mutation. I don't care about some random mutation. I need very, very, very specific mutations that would make me resistant to this particular virus. They would be different than the sort of mutations I will need to make me resistant to antibiotics and so on. So when I talk about probability of mutation, it's really shorthand notation for probability to get one of maybe a dozen different mutations, but super specific mutations in particular loci in my genome that turned a G into an A or whatever that made me resistant to this virus. Okay, so that's what I mean by mutation. It's a pooling together. Again, it's a coarse-grained model. I don't care about the details of which mutations and what it did. There are some mutations, and they have some probability to happen. The key point is this happens all the time, regardless of whether I've been exposed to the virus or not. Okay? So that's the model. A second thing that I'm assuming, which is not always true, is that if these mutations happened, they don't affect the doubling time of the bacteria. It's not always true. Sometimes, for cancer, I can have a mutation that would ultimately lead to cancer, but maybe it leads to a slower growth rate of the cell, so it doesn't survive for a long time, and usually it gets lost. Okay, so here I'm assuming what's called neutral mutation, that whether I got this mutation that makes me resistant to the virus or not doesn't affect much the doubling time of the cell. Okay, how do you see this in this tree that is drawn here from Wikipedia? How do I know that they're making this assumption? Exactly. When you look at these red, the doubling time is the same as here. The trees are, which brings me to another simplification. If you look at this tree, lineage tree, not to be confused with the phylogenetic tree that I showed you before. Okay, here it's the family tree of a single E. coli. What is unrealistic about it? It's a model. It's going to be a useful model. But what is completely unrealistic about this tree that we're going to revisit in the second topic of this mini course? Yes?
After the position, each bacteria takes the same exact.
Exactly. We're talking about variability, but we're neglecting another part of a feature of variability, which is the doubling times are variable in their own right? So this is sometimes called the synchronous model of division. You can do a simulation where you make it asynchronous. It won't change much. So that's a detail that we can ignore. Why? Because it would make for easier calculations. If we assume that all cells every exactly 20 minutes divide, calculations will be easier than if we get this crazy tree where some cells have 19 minutes and some cells have 22 minutes and so on. Okay? So sometimes let's say if I would do this today, I would maybe want to check in a simulation that the results are qualitatively the same when I make the model more realistic. But it's always good to do the calculations in some limits which are more tractable. So we're going to do the calculation in this limit of synchronous divisions and neutral effects of the mutation on the doubling time. Okay? So now we are pretty much prepared to calculate the variance to mean in this case. There's going to be some more complications ahead. But maybe I'm going to pause here and say calculating the entire distribution was super easy in the Lamarckian case. We just did it here and got the Poisson distribution. It's much, much harder in the scenario on the right. Okay? And in fact, there are papers to this day talking about features of this distribution, and I'll mention it either today or tomorrow. Actually, insights made by Mandelbrot, who worked on fractals, relating this distribution to power law tails and heavy tails and things like that. So it's a very rich problem mathematically, okay? And we're going to spend some time on its math. But there is something that's very easy to do. What's that? If you were, Max Delbruck and the year was not 1942, but 2025, okay? What could you do that you couldn't do back then? Simulate, okay? So it's a very powerful tool, and you shouldn't be afraid to use it, okay? So I'm going to show you next a code that basically solves this problem and finds within a simulation the distribution. It's more powerful than the calculations we'll do because it can give you the entire distribution, not just the variance to the mean. It can show you the results for a finite number of experiments that we'll see is important, right? The fact that they did 300 experiments, that number 300 also matters. It'll be different if they did 1,000 experiments. You're not in the limit where it doesn't matter the number of experiments. you can make a non-synchronous model very easily by making the cells divide at random times. You can make the mutation have a non-neutral effect. You can do all these things very easily in the simulation. Okay? But on the way, I want to show you how to do this simulation effectively. Okay, what do I mean by that? Probably if I tell most of you to make a simulation of this, you'll grow this tree in your computer, right? Is that a fair guess? You'll start with, say, a single cell. Let's say we start with a single cell. I want to start with a single cell and end with 10 to the 8 cells. Probably most of you will just grow a tree with 10 to the 8 leaves at the end, leaf cells at the end. Is that correct? Okay. So that's not so effective because you need 10 to the 8 to store 10 to the 8 variables and assign values to them and so on. There is a much better way to do it. So that's what I'm going to show you next. So to understand why there is a better way to do it, let's cut it by generation. Let's say I take the 7th generation of this tree. What do I care really about the 7th generation? What's the only information that's important to me? I don't care exactly which cell is red and which cell is blue if I follow this notation here. What do I care? What's that? Let's say I'm at the level 7. What information do I need to know about level 7 to go to level 8? So raise your hands so you don't speak at the same time. Yeah? I need a single number. I need to know how many red cells I have. I know that in level 1 there is 1 cell. In level 7 there's going to be 2 to the 7 cells. Okay, so overall there are 2 to the 7 cells. And if I know that there are 3 red cells, then I'm done. I know that there are 2 to the 7 minus 3 blue cells. And that's enough information to keep me going to the next level. So I need to store basically a single number, or 2 numbers if you want, how many red cells, how many blue cells. And then what happens? The red cells double, become twice the number. If I have 3 red cells, in the next level I'll have 6 red cells. Okay, why? Because we're neglecting back mutations that were mentioned before. It's negligible. We can forget about it. In principle, I could add that to the simulations too, but I don't need to. So the red cells double. And what happens to the blue cells? If I had 1,000 blue cells, in the next layer, how many blue cells do I have? Impossible to know. It's a random variable. Okay? The most is twice, but it can be less than that. So what I can do is I can double my blue cells. I have 2,000 blue cells. But out of these 2,000, what is the statistics of the red cells between again, I'm at some layer. Let's say I'm at layer 10. Layer 10, I have 2 to the 10, 1,024 blue cells. Great. I go to the next layer. I have 2,048 blue cells at most. Out of these 2,048, some potentially could become red cells. What's the distribution of the red cells going from this 1,024 to 2,048? Think Poisson. It's exactly the calculation we did now. Even though we're talking about the right model of mutations and so on, every replication event is independent. So the number of red cells in the next layer is going to be drawn from a Poisson distribution with a parameter which is 2,048 times the mutation rate. So I know also the parameter in my Poisson distribution. I can easily use MATLAB or your favorite software or do it by yourself to draw a random variable from a Poisson distribution with a parameter lambda. Then in our case, again, it's this 2,048 times the mutation rate. It's the chance that every replication event gives me a resistant virus. So that's a much, much, much more efficient way of doing this because it means that in the original experiment to go to 10 to the 8 cells, I had about 20 generations. 20 generations, all I need to do is generate 20 random variables out of my Poisson distribution, each time a different Poisson distribution. Yes? Let's assume, good question, let's assume that it doesn't really matter. It's a kind of detail that won't, so So let's assume that it's independent for each of them. Good question, but it kind of won't matter because the variance to mean is going to be of the order of hundreds. So this is a kind of detail that will affect the final results, but not qualitatively, but only quantitatively. OK, so here's how the code looks like. I took a mutation rate of the order of 10 to the minus 8 or 10 to the minus 7. I took 10,000 experiments. The code is so simple, you can run it for as many number of experiments. You can make this 300 to make it more similar to the original experiment. And now, for each experiment, I start with 0 mutations. And let's say I start with 10 cells, so 10 non-mutants. And every time I double my non-mutants and I double my mutants, okay, this is like the maximum number. But then I draw this Poisson from this potential number of non-mutants, the maximum number of non-mutants times the mutation rate, just like we said now. And now I make the total mutant number what it was before. The twice the previous non-mutant plus. This is the number of new mutants that emerged during this step. And the non-mutants is kind of the same. It's what I was supposed to have if there were no mutations minus the mutation events that happened. Okay? And now I kind of plot the histogram of the. So let's run it. Okay, so you can see it's sorry, I think I plotted it's not normalized my probability distribution, so ignore the actual scale, but it looks very similar to the experimental data quality. Yes?
Maybe it's just because it's a simulation, but why it is as if like you having this slope and then you rise up and then you have another slope?
Great question and great observation. So very happy with your questions. Keep them coming. And now, like I did it several times before, I will make the unfair thing with asking you, why do you think? Okay, so what feature in the model was unrealistic and led to exactly this observation? So think about, no, it's because we have a synchronous model. So what's going to, look at where you get your peaks, at 8. Okay? I have a synchronous model. It's pretty good for getting the overall structure and getting the variance to mean, but it means there is something special about getting 8 mutations, so because my divisions are so synchronous, I'm kind of favoring these powers of 2. So that's why I'm getting it. And if I will make the model asynchronous, it'll smear this effect out, and that'll be closer to experiments. Okay? So the important thing is if I look at the kind of variance to mean, I get 80. Okay? And if I do this again, I'll get a different number, 155. Okay? So for this parameter regime, the variance to mean, instead of being 1, is of the order of 100 or 200. Okay? And that's the thing we would like to understand next. Yes?
So the final, the both models will give me the same distribution. The Lamarckian.
No. So this is, OK, sorry. Let me try to explain it again. The fact that in the code I had this Poisson distribution is not for the number of survivors. It's for when you go from one layer to the next layer, given that I had a given number of non-mutants, what is the distribution of the number of mutations arising, new mutations arising in that layer? That is a Poisson. The number of survivors is very different than Poisson. You see it's very different than Poisson because the variance to mean, we just saw, it's 100. It's not 1. Okay?
So in the experiment, to get what model is the best?
What you measure in the experiment, you don't have access to what's happening during the growth process. You only have access to the endpoint. So the only thing you can ask about is the number of survivors. By the way, let me ask you this. If I take this tree of starting from 100 cells, ending with 10 to the 8 cells, and I look at the number of mutations that happened, it's hard to measure, but maybe possible with today's technology. We're actually in touch with a group in a different context to try to look at that. So let's say we do that. What do you expect the mutation number distribution to be? If I count every single mutation in this tree, what would be the distribution of the number of mutations? Number of survivors, because of this jackpot effect, very non-Poisson. But if I count individual mutations that happened, every time a cell mutates, a bacteria mutates, it rings a bell, and I can count how many times the bell rang. Okay? What would be the distribution of that? Not new mutations that happened. Maybe they give rise to a lot of mutants later on, but I'm counting only the mutation when it happens. The first mutation happens. What would be Poisson? Okay, so that's, I think, the source of the confusion. Mutations follow a Poisson distribution. Okay, but the number of survivors doesn't, because a single mutation, depending on when it happened, can have either a small outcome most of the time or a huge effect if it happens early on. Okay, there is one thing about the distribution of mutations. If P of M is the distribution of mutations, and it's supposed to be Poisson, so it has the same functional form as the one written here. So if I write M for mutations, this is the form of the distribution of mutations. There is one point in this distribution that is easy for me to measure. Which one is that? I said I can only count survivors. And survivors is different from mutations, except when? When do they when does the survivor give me also information about the mutations? Yes? Even simpler, if it doesn't happen at all. Okay? So the probability to have 0 survivors is 1-to-1 with the probability to have 0 mutations. Okay? Don't plug it into this because this is the wrong model. Okay? But if I look at the distribution of mutations, P of M is going to be e to the minus lambda, lambda to the m divided by m factorial, where lambda, what is lambda? I'm asking you a lot of questions, but I think it's good for your understanding. So lambda is the parameter for the Poisson, but what is it in this case? I need to count how many edges my tree has, because every edge corresponds to a potential mutations. So if you think about your tree, you end up with n cells. How many edges are there that lead to it? 2n. 2n, right? Because if you take approximately 2n, if you take this, right, I have 1, 2, 3, 4, 5, 6, 7, 8 cells at the end. Okay, but I'm counting edges. So there are 8 here, 4 here, 2 here, right? So if I take everything except for the last generation, I get n over 2, then plus n over 4, plus n over 8, and this geometric series converges to n, approximately, if I forget about this small wrinkle at the end. So I have 2n potential mutation event. So the parameter of the Poisson is 2 times n times mu. Okay? And now this is the distribution. Why is this useful? And it's useful to this day. It's useful because now P of m equals 0, okay, is e to the minus lambda, right? But that I can measure, because that's, what was it for the experiment I showed you? What is P of m equals 0? It was this 30-something% probability to have 0 survivors. 0 survivors is 1-to-1 with 0 mutations. So I can measure lambda by doing the Luria-Delbruck experiments many times and asking which fraction did I get 0. And that's a way to measure mutation rate. That's a good way to measure mutation rate. Okay? So we understand the structure of, this was a long answer to your question about Poisson distribution, where it appears. It appears in mutations, not in survivors, so don't be confused. now we have this model of this growing tree, and you can get mutation at every edge of this tree. And we want to know what is the variance and what is the mean of the number of mutants under the picture. The correct picture is that it's random mutations that drive evolution. So that's the calculations we're going to do next. And to summarize, so far we saw two models, Lamarckian that was easy to rule out because it predicts variance to mean of 1. And now we have the random mutation picture. We're going to see why it leads to variance to mean, which is large. We already saw it numerically in the simulation, and now we want to back it up by an analytical calculation. And all of this was done, except for the simulations, in their original paper from 1943 that resulted in a Nobel Prize, because they understood that random mutations drive evolution. And this is what I said opens now the door to understanding quantitatively evolution, now that we understand that it's random mutations that drive it, which you saw in the COVID is super relevant. And what we're going to do tomorrow is probably finish this calculation, but then talk more about microbial evolution and do some calculations related to, for example, things like fixation. I got a random mutation in a population. What is the chance that it overtakes my entire population? Which is something relevant. Again, if you think of COVID, what is the chance? Let's say Omicron has a 10% or 20% fitness advantage. What is the chance that it takes over the population? It's not 1. There is a good chance that it goes away because of stochasticity. So the stochasticity is very much at the heart of the topics we'll discuss this week. Okay, so let's talk about, can I erase this? Okay. So now we're going to calculate approximately the mean and variance and the key approximation we're going to make is a subtle one. Okay? The approximation we're going to make is that if I got a mutation here, let's say that this is this became from a blue cell here, a red cell here. So a mutation happens somewhere here. The chance that I get another mutation somewhere in the progeny of this cell is negligible. So I can forget about, I don't care if I get another mutation here, that's fine, because it doesn't affect it. But I want to kind of neglect the effect of sort of interference of two mutations happening in the same progeny. Okay? So let's first estimate what's the chance for this to happen. So let's say that after L generations, I got a mutation. So what's the chance of getting a mutation in the Lth generation? At the Lth generations, I have N0, the number I start with, times 2 to the L cells. Okay? And the chance of the mutations happening in each of them, let's call it P. So P times N0 times 2 to the L is the chance of a mutation occurring in the Lth generation. And if I want another mutation to occur somewhere here, I need to multiply by the probability times the number of replication events here. Now, if the number of total number of layers in the tree I call G, and like we said for the original experiment, G is of the order of 20, okay? Then the number of events here is 2 to the G minus L, maybe times another factor of 2 because of this property that we saw that the number of edges is twice the number of cells at the end. It's not so important, okay? Times P, okay? Because that's the chance of getting another mutation somewhere here. So we see that the relevant parameter, okay, 2 to the L times N0 times 2 to the G minus L, what is that? That's simply N0 times 2 to the G. What is N0 times 2 to the G? Total number of cells at the end. This is our, remember the key parameters, how many cells we begin with. The more important is how many cells we end with, this 10 to the 8. So this is the N final times p squared, we need this to be much smaller than 1 in order for us to be able to neglect having a mutation in the wake of another mutation. And that would essentially make our calculation more or less like the independent one that we had before. Okay? It means that if I have a mutation here, I can forget about the case. I can assume that I have no mutations happening again. Is this clear? Now, for the original experiment, n final times p was of order 1. You saw it in the simulation, right? I chose the number of cells to be 10 to the 8 of the order, and my p was of the order of 10 to the minus 8. So this thing is very well obeyed, right? This thing is of the order of 10 to the minus 8. It's really much smaller than 1, and this is an excellent approximation. Okay? How does this help me? Okay, so now I can calculate you will see in a second where it comes from, what it gives me. So if I look at the total number of survivors, because of this property, I can write, let's write Sn as the expected, is the number, it's a random variable, which is the number of survivors coming from mutation in nth generation. So I have a chance that I got a mutation in the first layer, and it led to this huge number of survivors, but it's a very small chance. I have a chance that I got a mutation on my 15th layer, much higher chance because I have many more cells, and that gives me many survivors, and so on and so forth. And I can say that my total number, S, is a sum over Sn. And I don't need to care about what if I got a mutation in the 15th layer, and then I got another mutation in the 17th layer. I'm kind of double counting. Well, this double counting is negligible based on this estimate. So that's where this property is entering into this subtle approximation I'm making here. And now things are very manageable. Because if I ask you, what is the probability to get a mutation in the nth layer? That's very easy. We just calculated it. I have n naught times 2 to the n times p. That's the probability of a mutation in the nth layer. Okay? How many survivors would that lead to? Okay, so if I look at Sn average, the expected number of survivors coming from mutations in the nth layer is simply the probability of getting a mutation in the nth layer times 2 to the g minus n. Is that clear? Because that's the number of progeny each of these has. Yes? No? So I plug it in and I get n naught 2 to the n p times 2 to the g minus n, which is simply, again, n naught 2 to the g is n final. So it's n final times p. Can anybody explain what we just calculated in words? What's special about this result? No, it's not a distribution. We just calculated an expectation value. Okay, good. You'll see it in the variance. You'll see it's it's not like the Bernoulli calculation. Because, I'll tell you why. Because Sn, each one of them, is indeed drawn from a Poisson distribution and so on, but its effect is it's different. I'm kind of summing these Poisson, but with different weights. So the number of survivors, like in this whole complicated, heavy-tailed, is nothing but a weighted sum of Poisson variable. But that's enough. The fact that they're weighted makes it complicated. That's the whole complication. So it looks like a detail, but it's not. It's a very good observation. OK, so I want you to kind of, again, stare at this result. This is the expected number of survivors coming from the n generation. OK, n generation. What do we get on the right side? Where is n? It's not there. What does it mean? I have 20 generations. What's their share in contributing to the number of survivors? Same. How can that be? Explain in words. Somebody explain in words why it's OK, why I didn't screw up the calculation, that every layer contributes the same to the final number of survivors on average, expected value. I took the expected value. Yes?
It's like if it appears in the first levels, it's less probable than it appears in a lower level. But it appears in the first level, it has many levels to go.
Perfect answer. OK? So this is in this calculation. The chance of it, the higher you go up the tree, the higher, exponentially, the higher the chance of getting mutation. But the effect of it is exponentially smaller. And these two effects cancel. So I have an equal contribution on average from getting a mutation, super rare to get a mutation in the first case. It's 10 to the minus 8. But then if I get it, I hit the jackpot, my entire tree is resistant. So I get a huge effect. And because of this cancellation, every layer contributes equally to the mean. Now, when you look at the variance, what's your intuition? What's going to happen then? Then this cancellation, we're just going to do the calculation now, is not going to happen. And the earlier you are, the bigger the effect you have on the variability. Okay? But the same logical calculation holds, but now no cancellations. The early generations contribute more to the second moment than the later ones. So it's kind of a coincidence that for the mean, the two effects cancel. And because they cancel in this thing, we find that S, the expected, this is the mean that we simulated and calculated. The mean is simply NF times P times what? The number of levels. We're in the synchronous model, so it's just 20 for that matter, so times G. So this is our first result, that the mean is final number times probability times G. Fine. Now we need to calculate the variance, OK? And we're almost, it's not going to be, the logic is the complicated thing about this calculation, not the actual technical details. So for the variance, I'm going to calculate, this is a random variable, right? The total number of survivals, S. Okay? And for the variance, I need to do S squared minus S average, S squared average minus S average squared. S average squared, check it, it's going to be negligible compared to S squared average. Okay? So actually, it's enough for me to calculate S squared average. And S squared average, it's taking the total number, okay, and squaring it. And now I can do the total number is the sum. N goes from 1 to G. Okay, and I have my N naught, 2 to the N P. Okay, that's the probability of getting a mutation in this particular layer. Okay, and I multiply it by 2 to the G minus N. So what's the key difference? I need to square this thing. This is the offspring of this particular mutation that happens somewhere in the tree, and I square it. So this is the calculation I have to do. And this is just a geometric series. You can all do this. And you find that this is approximately equal to P times N times 2 to the G. So we find that, indeed, because of this 2 to the G, this is a much bigger effect. This second moment is much larger than the first moment squared. and we find that the variance is approximately equal, or let's do the variance to the mean is approximately equal to p times n times 2 to the g, sorry, this should be n final, divided by p times n final times g, which is 2 to the g over g. G is 20, 2 to the g is 1,000,000, so we expect a variance of about 1,000,000 divided by 20. Okay? So I'm going to pause here and ask you a question. I'll go over this at the beginning of tomorrow's class because I felt like it was a bit quick. But I want to say this. In the simulation, we saw the variance to mean was about 100. We do this simple calculation, and the approximation should be a good one, and we get that the variance to mean is not 1, but it's actually 1,000,000 over 20. The problem is the opposite. It's too large compared to what we see in the simulation. Why do you think this is the case? Why is my calculation overestimating the variance to mean? It's a difficult question, which they addressed also in their 1943 paper. They understood this point. Yes? No, the model is fine. There's nothing wrong with the model. The problem is in this calculation, let's kind of look at this calculation. Do I really expect my experiments to give me n final p times g? We understood that every layer contributes equally to the mean. But what do I need in order to see the effect of the first layer? What must happen in some of my experiments? A mutation in the first experiment, in the first layer. How many experiments do you think I need to do to get a mutation in the first layer? 10 to the 8. Okay? But we only did 300 experiments. It means that it's unrealistic to expect us to sample mutations in the first layer, or second layer, or third layer. What is the first mutation? What is the first layer at which I realistically expect to observe a mutation giving contribution if I did 300 experiments? Well, I need n0, I need n0 times 2 to the effective generation number times p to be of the order times 300 to be of the order of 1. Because that's a probability that at least in one of my 300 experiments, I got this effect. This gives me a formula for what is the effective generation number at which I need to start doing all these sums. These calculations are still correct, but I shouldn't start them from the first generation because I have 0 practical chance of seeing a mutation there. I need to start them at some G effective, which is the first generation at which I have a real chance in my limited experiments number to see an effect. So this is a very subtle point, okay? But when you do that, you can account for the variance to mean not being 2 to the g over g, but being of the order of 100 or 200 as seen in the actual experiments. So this is a nice point because it means the number of experiments, you think 300 is essentially infinity. But really, to get to this limit, you need 10 to the 8 experiment, which of course is not realistic. So you have to account for the finite, even though it's a large thing, for the finite number of experiments you do. If you want to play with this, take the code, calculate the variance to mean when you have 300 experiments or 1,000,000 experiments, and you will see how this plays out and increases the variance to mean gradually as a function of the number of experiments. Okay, see you tomorrow. Yeah, of course.
Regarding the trees,
Yes.
if we just draw the tree